Welcome to the MS-Perspektive podcast, Perspective with a K as I'm German. I'm Nele von Horsten, MS patient, patient advocate and author. I'm bringing you interviews, inspiration and information about living with multiple sclerosis and show you ways how you can positively influence the disease and make the best out of your diagnosis. On ms-perspektive.com you will find the show notes, 11 free tips as a PDF and lots of other information. And now the show begins. Thanks for tuning in again and welcome to episode number 129. Hope for myelin repair. How stem cells could change the future of progressive MS with Dr. Luca Peruzzotti Jametti. And now enjoy the interview. Hello, Luca. It's such a pleasure to have you on the show, and thanks for having time for that interview today.
Well, thank you for having me. It's a pleasure to be here.
And before we actually start, it would be lovely if you could introduce yourself to the audience so they know who is with me on the podcast today and how you ended up in neural stem cell therapies research for multiple sclerosis.
Yes, no, absolutely. So my name is Luca, and I'm originally from Italy, where I studied my medical degree in neurology. So I'm a neurologist by training. And then I moved to the UK, where I completed my PhD and postdocs. And currently, I'm leading a lab that focuses on multiple sclerosis and neuroinflammation, and finding new ways to treat this disease. We came in contact with neurostem cells a long time ago, and already I was in Italy, working with my mentor, Stefano Pluchino. He was one of the leaders of neurostem cell transplantation in MS. And we worked a lot together, and we still work together, and it's been very fruitful for us and for patients.
Fantastic, yes. And we were lucky and had him for one session together with the International MS Management Master's Program when we were talking about innovative therapies. So I saw Stefano. So what drew your attention specifically to progressive MS and the challenges it presents?
Yeah, so during my training in neurology, I was actually working on a different disease, which was stroke. And there, I found out over the few years and with literature and studying that some of the pathophysiology that you find in stroke patients and in stroke lesions, it's kind of similar to what you would find in the progressive MS brain and spinal cord. So there are some issues related to how energy is missing inside the lesions, how there's a specific installment of a type of hypoxia that we call virtual hypoxia in these lesions that mimics very much what happens in stroke. So I think one of the interesting things that I brought to the table is that having all this experience in stroke research and in clinics, I looked at the disease in a slightly different way that it was normally looked at, and try to figure out how different metabolites and how stem cells could be working also, not in stroke, but also in progressive MS.
Okay, interesting. Can you tell us a bit your current research team and collaborations, such as with the Restore Consortium?
Yes, so we do a lot of work trying to figure out how neurostem cells can be used in patients with MS. As I said, the first publication that I actually made on neurostem cell therapy was dated back in 2008, and it was on stroke transplantation. And since then, we kind of like escalated a lot. There's been a lot of studies using neurostem cells as a way to not only control the inflamed environment within lesions, but also ultimately to replace damaged cells. And with going up from doing preclinical research into mouse models, Stefan has done a lot of work also on non-human primates. And then finally, we were able to have a Phase-1 clinical trial with the transplantation of neurostem cells in progressive MS patients, which was published in Cell Stem Cell in 2023. And we're slowly building on that. And the idea is to move forward to a phase 2 soon. The Progressive MS Alliance has been instrumental in this. They've been helping us both financially as well as trying to figure out things as we go along. And we have a grant to trying to build this consortium for a phase 2 trial.
Fantastic. Yes, I have to invite them to the podcast, the Progressive Alliance. Very interesting what they are all doing. I think we need to explain a little bit before we dive deeper into the research. What exactly happens in the brain during a relapse and during PIRA, the so-called progression independent of relapse activity?
Yes, so I think that's the key question, right? Because we have, thanks to the work of many people, so many treatments now for the early relapsing remitting phase. And in that phase, what happens is that the immune system, which should, defend ourselves from pathogens, just decides to attack the central nervous system. And there's a lot of these immune cells coming from the periphery, from the body, and entering into the brain. So all the majority of the therapies that we have are trying to either limit the entry of immune cells into the brain or reducing the number of cells that get into the brain so that we can prevent these relapses from happening. And I have to say that this is a cornerstone of treatment. The more we prevent relapses, the more we can salvage the brain from damage. Now, unfortunately, in patients, there is a progression that ensues independently of relapses. So after they had their disease for a period of time, they start, unfortunately, to develop a progressive disability that is independent of these attacks coming from the periphery needed by the immune system into the brain. So what is happening, we think that there's a kind of switch of this immune activation. Instead of being driven mostly by the periphery, there's an inflammation that becomes compartmentalized within the brain. So a chronic state of inflammation that is independent from these attacks coming from the periphery. And the cells that we're studying now are the microglia, which are the so-called major immune cells resident in the brain. And what we see in these patients is that there's a lot of these microglia become activated and throughout the brain and also in specific lesions. And these cells are thought to mediate this progression independent of relapses activity that we see in patients. So the idea is to find new ways, which we're currently lacking, to not interfere with the immune system in the periphery, but rather interfere with these microglia in the brain.
Yeah, fantastic. So what role does myelin play in nerve function and what are the consequences when it's damaged in MS?
Yes. So, as you know, our nerves are kind of like wires, and they transmit the information from the brain to the periphery and from the periphery to the brain. And what happens is that this information, because it has to go such a long length, it needs to be fast. So one way would be to have huge axons, which is obviously not doable. But the other way is instead to coat these axons with myelin. And myelin works as an insulation that helps to speed up this information coming from the brain to the periphery and vice versa, so that we are fast and we can do things correctly. Now, what happens in MS, as you know, is that unfortunately myelin becomes the target of this immune attack to the brain, and this coating of the nerves, unfortunately, is lost. And so that leads to other symptoms that could be transitory, especially if the coating is kind of like damaged, especially by nitric oxide or other things that block locally the conduction. And then these symptoms can go away if the inflammation is treated. For example, a lot of patients during relapses take steroids, which help them to get better much faster. But in many cases, the constant damage to the myelin causes also the axons to die, and not new myelin can be formed efficiently, at least in the human brain, and therefore there's a growing number of lesions and disability that appear in patients.
Yeah. So why is the repair of myelin so limited in progressive MS? Why is that such a problem?
Yes, there's been many ideas behind this. So first of all, it seems to be something related to the human brain. The human brain has a lot of plasticity, especially in the early phases, up to when the brain becomes fully myelinated around your 20s and 20s. And this is meant to help you be able to, grasp new concepts and develop new ways to do things and new functions while you're growing. And as a matter of fact, when there are diseases that affect myelin early on in patients, the repair abilities are much better. Now, in progressive MS, as you know, most of the patients are actually in their 50s, 60s, and the regenerating ability of these patients is dramatically reduced, not only because they have progressive MS, but also because they're older, and therefore it is difficult for them to repair. So that's the first thing. And then the second thing, it seems to be something very, very intrinsic to the human brain. We're fundamentally different from small rodents or other animals in which there's a lot of these precursor cells that are able to remyelinate locally if there's a damage. In patients, this doesn't seem to be happening as often as we would like to.
Okay. And what kinds of cells are usually responsible for remyelination, and what goes wrong with them in MS?
Yes, so this is the work that another eminent scientist in Cambridge has done for many, many years, Robin Franklin. And he has been studying how remyelination happens in demyelinating lesions and MS and other diseases. And essentially, there are some precursor cells, which we call OPCs, or oligodendrocyte precursor cells. These are cells that are able to migrate, locate within lesions, and then differentiate and actually proliferate also, but not differentiate into oligodendrocytes. And oligodendrocytes are the final cells that are able to myelinate axons. Now, in MS, all this very complicated process, which requires the cells to go there, proliferate, differentiate, and sheet the axons, is broken at several levels. So there's a block of migration, and there's a block of proliferation and myelination. And it's very complicated, but there are several players there. There's obviously the component that is intrinsic to the oligodendrocytes transforming into the OPCs transforming into oligodendrocytes, but it's also extrinsic cues. For example, in the lesion, they become more stiff, so the stiffness of the lesion plays a key role there. The axons also don't send the right signals, and of course, if there's ongoing inflammation, also these cells become less prone to differentiate.
Yeah. Yeah, we saw nice pictures when we studied with Professor Stadelmann-Nessler, where they are trying to remyelinate, but destruction is still going on, and then
Yeah,
they destroy what already was repaired. And yeah.
yeah, this is exactly actually what we see with our neural stem cells when we transplant them. So we can talk about more about it later.
Yeah. So can we say that neurodegeneration in MS is preventable or even reversible if we manage to restore the myelin?
Preventable neurodegeneration, if we could restore myelin efficiently, yes. As a matter of fact, there are many trials that are being carried out worldwide, and in Cambridge, the leader here is Alistair Coles, who's been doing all these trials to try to prevent demyelination and increase remyelination. And the thought behind these trials is to do it earlier on. So if you could do this early on in the disease, not wait for the progressive phase to ensue, then you could help the resilience of the brain and avoid neurodegeneration. So I think that's the common thinking now. In terms of regeneration, that's the jury is still open there.
Mhm. Okay. So now we can, I think, dive deeper with all that knowledge you just provided us. What are neural stem cells, the NSCs, and how are induced neural stem cells, iNSCs, different?
Yes, so that's a key question. So let's start with the neurostem cells. So I think we have to describe pretty much why we're focusing on neurostem cells and not other stem cells. And so there are many reasons, but essentially what a stem cell does is that it's able to proliferate. So you can maintain it in culture long term, and you can expand it. And the most important thing is that the cells are able also to differentiate into other cells. Now, you probably know about hematopoietic stem cells, mesenchymal stem cells, neural stem cells. So these are different lineages that are able to, once they differentiate, to give rise to different cell types. Now, neural stem cells are the stem cells that are in the brain. So by default, their intrinsic ability is to give rise to neural cells. So both neurons, as well as oligodendrocytes, as well as astrocytes. These are the major three cell types. And essentially, the main advantage of these cells is that they're brain specific. So if we could harness them and transplant them and make them able to differentiate into neural cells, we could replace part of the cells that are lost. So this is the main reason why we work with neural stem cells. Now, there are some problems, obviously, because it's not as easy as we are discussing now to get neural stem cells for transplantation. Most of the studies, including our own study, the Phase-1 that I mentioned before, uses neural stem cells derived from fetal or embryonic sources. So obviously these are allogeneic transplants. So you have a donor, which is a fetus, and then you have your neural stem cells. You make this advanced medicinal product made of neural stem cells, and then you use it for many patients. But there are, as you know, incompatibilities related to the graft. Like any graft that you will receive from another person would require some sort of immunosuppression. And that is okay for multiple sclerosis, especially because many of the therapies that we use are immunosuppressive therapies. But long-term immunosuppression may be not, usable and easy to do in patients with progressive and advanced multiple sclerosis. So the idea would be then to obtain these neurostem cells, which would be different from each patient to each patient. So in a few years ago, Yamanaka, who received the Nobel Prize for Medicine, uncovered these transcription factors, which are kind of like instructions inside cells that allow cells to de-differentiate from being skin cells, for example, which are fibroblasts, into stem cells. And so the induced neural stem cells that we're using are a variation of this technology in which we use skin fibroblasts from mice or from patients, and we differentiate them to acquire a neural stem cell-like features. And we call them induced neural stem cells because they're induced in vitro. And these cells have the great advantage of being patient specific. And so there's no risk for immunorejection at any point.
Very nice. I just remember that movie, I think it was with Brad Pitt, something Meeting Joe Black or so, where he transformed from an old man back to a baby. He was going that other way around.
Oh, that's Benjamin Button.
Benjamin Button, sorry. Okay, was wrong. But yeah, it was an interesting movie. Okay, so you mentioned the paracrine effect. Can you explain what that means in simple terms and how it contributes to healing in the brain?
Yes. So essentially, when the first experiments were done with the neural stem cell transplantation, the idea was primarily the one to replace the tissue. Because of the experience in other fields of medicine, we found out, and other people found out, that if you transplant these cells in an inflamed environment, their propensity to differentiate is actually not great. But it's not that they're useless; they actually do other things. So they're able to get into the brain. They are equipped with a series of sensors, which are these receptors that are present on their surface that, interestingly enough, recognize a lot of immune molecules. and they're able to locate specifically within inflammatory lesions. And when they get there, they secrete these other molecules, which are anti-inflammatory in some cases. They promote regeneration in other cases. And as we demonstrated back in 2018, they can also modify locally the metabolic environment to get rid of metabolites that are known to have pro-inflammatory functions. And so they create a more an environment that is more prone to regeneration, and they block inflammation locally. And that's the paracrine effect that we have seen, and many other people have seen also when transplant-induced neural stem cells.
Okay, nice. In your studies, NSCs produced new myelin and influenced immune responses. Which of these mechanisms is more promising for future therapies?
Ideally, both. We certainly have data that is very convincing about the reduction of immune responses, which is, I think, very great, and it's solid. It's 20 more years of research. In terms of replacing myelin, it's more complicated because it depends very much on the model that we use. It appears that we really have to control inflammation in the brain before these cells can differentiate. Um, and going to the matter, for example, one of the papers that we recently published in Brain this year, we had to use a different model of MS to show that these cells are really able to replace lost myelin. And this model is a model that doesn't have a lot of inflammation. If we use other models which are overtly inflammatory, these cells are not able to differentiate as much.
Okay. And just for the listeners who are not maybe that deep into the topic, the problem is there is no multiple sclerosis in animals. So there are different types of animal models, but unfortunately they can't be combined. So researchers like you have to look into different aspects of the disease, but in humans it's still a bit different, right? I
Yeah,
think that's important for everybody
absolutely.
to know.
It's a huge limitation of current research. The fact that we don't have multiple sclerosis doesn't happen in mice, and we have to kind of like model the disease. These are mouse models. They mimic part of the complex pathophysiology of the disease. So there are some that are more established for inflammation, some that are more established for myelin damage and regeneration, and we kind of like have to use two or three of them sometimes to make sure that a therapy will work eventually in humans.
Yeah, thank you. How safe is the transplantation of human cells based on your current research?
Safe. So the cell stem cell paper in 2023, and there's also another group from Milan who published a Nature Medicine paper using neural stem cells transplantation in MS patients. And in both cases, there were no significant adverse effects. In their case, the administration was done through a therapeutic lumbar puncture, which worked in delivering the cells. In our case, we were thinking that it was much better to get more closer to the brain. And so we even did a more invasive approach with neurosurgery and intraventricular administration. And in both cases, despite the patients were disabled, and in our case, the EDSS of these patients, which is a measure of disability, was quite high. They still didn't have major adverse effects from the transplantation procedure or from the transplant itself, which I think it's very reassuring, and that's why we're moving forward with the phase 2.
Fantastic. So let's move on to the realistic perspectives and having a look into the future. How far are we from seeing the first large clinical trials for neural stem cell therapies in MS?
It's a good
I know it's a hard question. Sorry for that. We want to know, you know?
Absolutely. If I have to give you a number, 5 to 10 years, but it could be sooner, it could be later. Unfortunately, we have to do things. So one of the first things that we learn in medicine is that first we don't have to create harm to patients. And so we really have to go through these therapies in the proper way, going through all the phases of clinical trials. That you heard me saying phase 1 multiple times. And that's the most important phase, which kind of like looks at toxicity. It doesn't look at any efficacy, but just toxicity and possibility of damage. So if we clear that phase, then you have to go into other studies that include the dose escalation. In our study, in the phase 1, we already had different doses, but obviously this is something that needs to be further refined. And then you can go into studies of efficacy, which initially are small, and then if they catch the eye of pharma companies or other support from societies, they become bigger trials. So it's a long way. I think both the patients as well as the researchers, such as myself, are very, very, very much looking forward to do this, and we are really working around the clock to make this happen. But unfortunately, it's biology, it's research, so we just need to be sure that we're doing the right things. The risk is that if things are not doing in the proper way, we just go quickly into things and then we get a negative result or even worse, adverse effects, then the entire field is killed and this therapy will never get into patients.
Yeah, yeah. Yeah, and thank you for giving a bit an inside look. I will have another episode on that. In October, I will finalize my UPATI program, where it's all about clinical studies, and I will have some more episodes here. But yeah, hopefully it will go smooth, but maybe some back and forth steps are normal, I guess.
Yes.
What are the biggest hurdles still standing in the way, scientific, regulatory, or practical?
I would say a bit of everything. So the regulatory part is very difficult. Obviously, we are kind of like lucky in the UK. We have the MHRA, and they're very proactive in bringing new therapies into the clinical realm. And we in Cambridge, we are like the common approach to this is to interface with them early on so that we know exactly what needs to be produced so that we can go into the clinic as soon as possible. So that's helpful, but it is a lot of work. And in terms of the practical things, I have to say that uh, especially with the phase 1 clinical trial that we did with Professor Vescovi, the advanced medicinal product is there. So it's just a matter of making sure that it can be shipped. If we're talking about a multicentric clinical trial, it can be shipped in between the different centers, and the thawing and the processing of the cells is consistent in all the centers, and so is the injection. So there's a lot of work, but I think we can overcome many of these things. And going back to your previous question, I have here, and maybe I can get it.
People can't see it, but I see you grabbing somewhere.
Yeah, I put it here because it's a reminder of how long things take
Mhm.
get into the clinical research and clinical practice. So Alistair Coles, whom I mentioned before, is known for his work in immunology, and especially in one of the therapies that we use in MS, which is Campath, dialemtuzumab. Originally, it was called Campath because it was developed in Cambridge pathology. And so I have here this one. I don't know if you can see it. So it's a very nice timeline between when the antibody was discovered and when NICE, which is our approval body, approved it for MS. So it was first discovered in 1980, and and it was then approved from NICE in 2014. So that gives you kind of like an idea of how much work we have to do to bring a biologic into patients. So you can imagine how difficult it is with cells, which are by default more heterogeneous than a monoclonal antibody.
But yeah, you normally don't cause in 40% of patients another immune disease.
Yeah, yeah,
Alemtuzumab. So,
that's
but
absolutely.
of course, it's a very effective therapy. For those one who have a very aggressive type of
Yeah.
MS, it's a very good choice. Uh, yeah. It takes long. We had that in the multiple sclerosis management program with the different therapies. It takes so long. But still, it's important to have some hope and to hear about things are moving, even though it takes longer than sometimes
But I think we don't have to see it as as a linear structure in which it takes so long, but more of like multiple lines working at the same time. So we're working on neural stem cells, but there are other people working on other approaches. And so at some point we're stacked and they all will come out as they did in the past 10, 15 years, new therapies every time. And we don't have to wait for neural stem cells. There will be another one coming out soon. So there's a lot of research and buzz around multiple sclerosis, and we're really struggling to get these therapies out, and a lot of people are working on it. And so I want to give like a positive outcome, because I didn't want to, and if it is not neural stem cells now, other therapies will come out, and then eventually neural stem cells. So it's work in progress.
Yeah, absolutely. The next big one will be the BTK inhibitors, but there are more interesting therapies in phase 3 already. So yes, always good. And it's an interesting field for researchers, so that's great for us. Could the therapy be used for all people with MS, or mainly for those with progressive forms? And that was a question coming from two gentlemen just a few days ago. Would it be possible to do it after an aHSCT, as far as you know, as of now?
Yes. So, terms of could be used, the answer is yes. I think the real question should be, is it worth using it? And so I think the answer is maybe not. In the early phases of MS, I think we have so many therapies now that are so effective in preventing relapses, going from the most effective ones to the least ones, depending on disease activity, that probably you don't want to use it early on in the disease. However, when patients start that progression independent of relapses activity, when they start accumulating disability, that's when we don't have a treatment, we don't have effective therapies, and that's where neural stem cells will likely be used the most. In terms of combination with other therapies, like hematopoietic stem cell transplantation, I think that's something that can be done. Obviously, not at the same time. You probably need to do your HST transplant, and then after a period of time, if you go into progression, then probably have neural stem cell transplantation.
Yeah, yes, it was a question from two people with kind of like primary progressive MS who had the aHSCT and were not seeing good progress and hoping, of course, for then remyelination procedures. How do you see stem cell therapy in relation to other new therapeutic strategies, such as targeting mitochondrial metabolism?
Yes, that's a great question. Actually, my lab is mostly focusing on the second one,
Mhm.
on the metabolism. And all the work that I've been explaining is mostly done in collaboration with my mentor, Stefano. And so we obviously keep pushing the neural stem cells because we've been working on it for such a long time. We really believe that it could bring benefit to patients, and we're closer to the clinic. Well, the work that my lab does is more experimental at the moment. So we're still at the beginning. It's a new field. And, I was talking about, stacking therapies one on top of the other one. We're starting with this one. So we hope that in 10, 15 years, we'll be ready with the new biologics that work on this new mechanism of disease. I can give you more details on what we do, but essentially that's our plan for the future. Looking at bioenergetics within lesions and help kind of like heal these lesions and rejuvenate a little bit the brain, make it a little bit more fit, which is also one of the problems with the progressive MS.
Yeah, fantastic. So, you mentioned there are quite a lot of different types of therapies. You would say it's realistic that we will see a therapy that not only slows but reverses disability into the next 10 to 15 years with all that researchers around the globe working together and sharing knowledge and using data and using real world data and things like that?
And I'm a glass half full person, so I would say yes.
That's good. Great. Okay, so let's come to the quickfire Q&A session. Complete the sentence. For me, multiple sclerosis is?
Fascinating.
What development would you like to see in the field of MS in the next 5 years?
Slowing down progression efficiently.
Mhm. What message would you like to share with people living with progressive MS who may feel left behind by current therapies?
We're here for you, always.
How and where can interested people follow your research activities?
They can. We have a progressive MS Facebook page for Cambridge. Our lab is very active on Blue Sky, and so that's social media, and of course there's a lot going on the Cambridge websites as well. So many ways to get in contact, and obviously I'm very happy to answer to questions even offline. Just send me an email.
Fantastic. Great. And I will put all these links in the show notes and in the blog article. Luca, thank you very much for giving such an interesting insight into your research topics. And of course, thank you to everybody who is working in the field in Cambridge and elsewhere, because it's always one person I speak to, but there are always teams behind that and cooperation going on. And I'm looking forward to ACTRIMS. I won't join this time in person, just online. But I guess there will be interesting topics be discussed, and I will share them here on the podcast. So thank you very, very much.
Thank you for having me, and it was a pleasure. And I would like to say, goodbye to all the people listening and all the people with MS. And we're really doing a lot of work. We have a lot of public engagement activities as well in Cambridge, and I think that what you're doing with your blog is very much important to get the word out, so that people don't feel like they're left behind, but actually they understand that there's a lot going on.
Yeah.
So thank you.
Communicating the scientific outcomes, that's important. Yeah. Thank you. Bye-bye.
Take care.
I hope you enjoyed the interview, learned something new, and got inspired that there are really fantastic researchers out there who are committed to help you, to help me, to help all of us. By the way, next time I will be having an interview with another MS patient, and the topic will be on living with MS abroad. This is a conversation with Robert Joyce on research, resilience, and life in Spain. Robert is originally from Ireland and is now living in Spain, and he is a very engaged patient advocate, and he will talk about all this next time. So please tune in. Please stay as positive and healthy as possible, and let others know who might be interested in the podcast that it is existing. If you are on a platform where you can rate it, please do so. And of course, comment on social media so others get to know this podcast. And by this, see you next time. Bye. Thank you for listening today. I hope you found it helpful. You may also be interested in the previously published episodes and posts, which you can find at ms-perspektive.com/podcast. Perspective with a K. You can also get a free PDF with 11 tips for your positive impact on MS at ms-perspektive.com/newsletter. If you'd like to get in touch, you can reach me at Nele at ms-perspektive.com or on social media. See you next time.